Hello and welcome to the IEDB Virtual User Workshop for 2022. My name is Nina Blajeska and I'm the Project Manager of the IEDB. I'd like to thank you all for joining us today from whatever part of the world you are in for day one of our 3-day event. I'll be giving you a brief overview before we jump into our presentations for today. So why do we host a user workshop every year for the IEDB? Well, it enables us to engage with our global user community, and global it is because we have people registered from over 60 countries this year. This year, we have our first 3-day event, previously 2 days, where we'll cover the IEDB database and tools, providing an overview, also some research examples and other specialized topics which are new for day 3. It also gives us the opportunity to improve our resources through user feedback, which we'll gain directly from this webinar. It allows us to answer user questions to help facilitate learning, and this will be done through our Q&A functionality within the Zoom webinar. And finally, it enables us to share ideas to further scientific research, because what is science if not collaboration? As some of you may know, we've gone from this, so previously an in-person workshop format on a slightly smaller scale due to size restrictions, to this. So we obviously know what happened in 2020, and at that time we pivoted to the virtual workshop format, and we had greatly successful events, so we continued that format for 2021 and 2022. Of course, so with these new challenges, it's enabled us to create new opportunities. So we are coming to you once again in a completely virtual format from the comfort of your own home. This year, we've also registered and approved over 550 attendees. So breaking all of our records once again, this is our largest workshop ever. And finally, we're bringing new presentation topics to you on novel research and new tools as well. So over the next couple of days, we hope that you engage with our presenters and enjoy the materials that they're presenting to you, and most importantly, learn something new that you can take away and apply to your own research. You'll be hearing from a host of wonderful people over these 3 days. Firstly, Dr. Alessandro Sette, our principal investigator of the IEDB, and Dr. Bjorn Peters, our co-PI. You'll also hear from Dr. Randy Vida, our lead ontology and quality manager, and Dr. Sydney Fitzpatrick, our scientific curator. You'll also hear from Kelly Wheeler, our senior software engineer, and 2 of our instructors at LJI, Dr. Alba Grafoni and Dr. Zeynep Kozaloglu Yaltsin. You'll also hear from Dr. Rafael Trevisani, Dr. Mahita Jajapu, and Dr. Marcus Mendes, 3 of our bioinformatics postdoctoral researchers. Daniel Marama, one of our bioinformatics research technicians, will also be presenting, Dr. Jason Greenbaum, our bioinformatics core director, and Dr. Paolo Marcatilli, one of our colleagues from the Denmark Technical University. So that was a team in the IEDB, but we also think it's important that you hear from other NIH resources. So you'll be hearing from the Los Alamos National Laboratory, namely Dr. Elizabeth Sharon Fung and Dr. Jennifer Mackey on day 3 as well. So the overall structure of the workshop is very deliberate. Day 1 gives you a process overview, and we really focus on the database specifically. Then day 2, we will look at the counterpart to the IEDB database, which is the analysis resource, namely the processing and prediction tools. Day 3, now this is our newest day, we'll be covering specialized topics. So these topics will range from 3D structure analysis to cancer resources and also HIV databases. Now, of course, these 3 days, you'll have a series of presentations from all of those people that I've just mentioned. But we'll also be responding to questions in the multiple Q&A sessions throughout that agenda. Now, we request that you use the Q&A function in the webinar, just shown in this image here. So feel free to pop in your questions and we will get to those in the Q&A sections. There will be one break on each day in the agenda, roughly 25 minutes. and we will keep you updated on those timings in the chat. So we do request that you post your questions in the Q&A function, but we will keep you updated in the chat on break times, some links to extra resources, and then of course our important surveys for each day. Lastly, Zoom webinar also has the ability to show you live transcripts or subtitles in real time. So that has been enabled. So feel free to select the different options and you can show subtitles on your screen as well. So now looking at day one as an overview, shortly I'll be handing it over to Dr. Alessandro Sette to give you a database overview. And then Bjorn will be presenting the analysis resource overview, so that counterpart presentation. After the Q&A, we'll hand it over to Randy and Sydney to show you how to access the data, query, reporting, and examples. After that, Kelly will be presenting some new database features for you, the custom exports. After that Q&A, we will have our break where we'll share with you the resources and our survey. Then you'll be hearing from Alba on how to use the IEDB in your own research, and she'll be showing you examples on orthopox and SARS-CoV-2 data, which will be great. Finally, you'll hear from Alex once again on the analysis tools. After that, we'll finish up with our last Q&A for the day, some closing remarks and our survey, and then we'll wrap up at 12 p.m. Pacific time. A couple of other event logistics to be aware of. So you will receive a post-event email next week. In that email, you will have the workshop recording shared with you. So we'll be uploading that to YouTube. All presentations will be shared with you as well. Participation certificates will be requested upon, provided upon request, and we will be sending a post-event feedback survey in that email as well, in addition to the daily surveys that you'll be receiving. And please just be aware, if you are trying to use the IEDB over the next few days, there may be some slowness due to the increased traffic from the workshop. Lastly, you might know, but we are on Twitter. Please do follow us at IEDB_ if you're not already. We'll be sharing some resources from the workshop on Twitter, so we'd love you to do the same. Please share anything that you've learned or found interesting. Tag us at IEDB_ and use the hashtag IEDBUW2022. We can't wait to see what you'll share with us. And now, without further ado, I would like to introduce Dr. Alessandro Sette to present the database overview. Thanks very much, Alex.
Thank you, Nina. Let me share my screen and let me put it on presentation mode. Hopefully you can see that. Yes, so what is the IDB? Let's go into this, a little bit of an overview. This slide is somewhat redundant with what Nina has already outlined. Basically, what we want is, on the bottom, our main goal is to enable you to use the IDB, facilitate the use of the IDB by describing the different features and so forth. And this is important to us because obviously, what is a database if nobody uses it? Conversely, we also get a lot of it out of this workshop, because what we hear from you is crucial for us in order to make the IDB better, learn from you what works, what doesn't work, what messages comes across and what messages don't come across because they're maybe not intuitively apparent what we're doing. And over the years, in fact, the IDB has improved a lot, really relying on these workshops as one of the main sources of feedback. So what though is the IDB? While you will hear from Bjorn about the analysis resource components, the IDB is also and primarily a database of information regarding epitopes. And this is all about experimentally defined data. So the database doesn't store any predictions, it's just experimental data generated by labs all over the world. And the field is very broad. We essentially curate everything from allergens to infectious disease, autoimmune diseases, transplantations. In the third day, you will hear about the sister database, the CEDAR, which curates specifically cancer epitope data. And also the Los Alamos database is a fantastic resource for what relates to HIV epitope data. The data we curate is from humans, from mice, from non-human primates, pretty much any species for which there is data relating to epitopes and associated assays we curate. The IDB currently contains data from about 2 million different structures and epitopes defined in about 6 million different assays. And as I will elaborate more in a second, the IDB is really a database of assays and experimental data, and the user has the capacity to drill down and really get to the nuts and bolts of what are the experiments that underline someone saying this is an epitope. This information is derived from over 20,000 different scientific references published in peer-reviewed journals, and also, as I will explain, to a certain extent, to a large extent, also from data derived from contracts that have been funded from NIAID and other sources to generate large-scale epitope information. So, immune epitope database, but what's an epitope? What's an immune epitope? And so we had to define to what level of granularity we consider that an epitope has been defined, and we set this criteria, somewhat arbitrary, but we say that in the case of a linear peptide, it has to be 50 amino acids or less in length. So if there is some data of someone that they made an antibody that reacts with the surface antigen of hepatitis B, well, that's not an epitope, but that's an antigen, it's a protein. So it has to be defined to a certain degree, and we set this bar at 50 amino acids. And the peptide or the epitope has to be tested as a immunogen antigen. So it's fine if someone, for example, has immunized with a whole protein, but then used defined epitopes to detect responses to those defined epitope, or conversely, it's fine if someone perhaps used a defined epitope to immunize some mice and then read the response in terms of is the response capable of recognizing, for example, infected target cells. We curate, particularly in the case of antibodies, it's rather frequent that the epitope is not defined more accurately by a large linear sequence, but rather from a number of different discontinuous residues that are brought together conformationally to form an epitope recognized by antibodies, and we curate that. We also curate non-peptidic epitopes, less than 5,000 Dalton in size. And we also require, in order for an experiment related to an epitope, that there is some minimal information, such as what's the sequence or what's the structure of the epitope, and what was the outcome? Was there an experiment that was run? What was the outcome of the experiment? And was this in a mouse or in a macaque and so forth? Now, the challenge that the curators encounter in curating epitope data, and we have encountered more generally in structuring the database, is that scientific reports have a lot of information that is dispersed throughout the scientific paper. So, for example, you may have, in this particular case, a smallpox reference in A201 in the concentrate 201, HLA molecules. And so the experiment is actually described in figures and tables in the paper. What was actually done is often described in the method section, which is a different part of the paper, and the interpretation and the outcome is described in the results section. So the curator has to extract this information that is contained in the paper and put it in a format which is curatable and is understandable from in terms of a computer format. So here you see that eventually the information contained in this scientific published peer review journal article is distilled to say what was the actual structure of the epitopes, what was derived from, what was the context, was an immunization, and that was an administration by scarification, and then the response was read using the defined epitope in vitro in an ELISpot measuring interferon gamma and so forth. So the database is really, as I was mentioning, essentially a database of experiments where we curate each single kernel of information, each single experiments. And so as I was showing, there may be a reference that might have different epitopes, and the different epitopes may be tested in different assays with this information or multiple assays. And of course, to make the things even more complicated, the same epitope may be tested in multiple different references from different laboratories. A key element of the database is the reliance on collaboration with other ontologies. And so we rely on other ontologies and other resources that maintain different domains of knowledge to have standardized nomenclature, definitions, synonyms, and so forth, which is very important because this makes the resource truly interoperable with other resources and speaking common scientific language. There are additional benefits because, of course, we don't want to maintain the nomenclature for the whole universe. And so to rely on external well-established resources makes the curation easier, makes it easier for the user to find the information because we're speaking a common language, and ensures consistency and accuracy. And also we can establish validation rules that allows to find errors and prevent errors in the first place. So for example, in the case of the taxonomy, we rely on the NCBI taxonomy, and this is behind the scenes is what we use to categorize the taxonomic source of the different epitopes, or in the case of nomenclature relating to different proteins, of course, there is GenBank and UniProt that allow us to link our records and link the sequences from which the epitopes are derived to a very well-established and maintained series of ontologies. So how do we go about curating the information? So first thing is we have to find what are references that are potentially relevant and contain epitope related assays. So we have a very broad query that we run every other week, mining the PubMed and PDB for records that are potentially curatable. And so this is by design very, very broad. We try to capture everything potentially relevant. And by doing so, we have over years looked at about 250,000 different articles. This information then is entered into a classifier that we have developed rather rigorously, and actually there is a series of peer-reviewed publications describing how we develop this automated classifier that recognizes and selects the references that are likely to contain epitope information, and then it cuts it down to about 150,000 records. And then we actually review every single abstract manually of the ones that are sieved through the PubMed query in the classifier. And this stage zeroes in on likely credible papers. About 40,000 survive at this point. And then this is now passed on to a team of PhD level curators that look at the paper, not just as the abstract, look at the paper in its entirety, and determines whether it really does contain curatable information. At the end of the day, as I was saying, over the years, we curated over 20,000, 23,000 different peer review reports. And here you see how it went throughout the years. There was a number of years where it was a very valiant effort to curate the literature, both being curating new references, but also going back in time. And we caught up in about 2011. And from there, we've been more in steady state with a gentler slope. What are the references derived from that are curated in the IDB? You see the about half, a little bit over half, are from infectious diseases. And then there is about 10% from allergy and quarter autoimmunity, relatively fewer transplantation, and then others, other references that are not easily classifiable in these different things, and maybe, for example, a monoclonal antibody made in mouse against a human interleukin, or things like that. So the curation process is been going on over the years, involves a team of curators. It's very important that there are consistently defined and applied rules, so that what is curated by one curator is corresponds to the same thing if a different curator is curating the same, where to curate the same references. And we have a formally defined set of guidelines. There's a curation manual that is actually accessible to everybody to really define in very much detail how do we curate things. We have external immunological experts that help us also pressure test our definitions. Also, the curation has evolved over time as the scientific literature has evolved over time. There is an effort of recurating to make sure that things are up to date and consistent. We rely on your feedback. And also, there are automatic validation rules that prevent error from creeping in the database as much as possible. I mentioned earlier on that the IDB not only hosts data from the scientific literature, peer-reviewed journal, but also hosts data from NIAID epitope discovery contracts. And on a case-by-case other instances in which someone has a lot of data that wants to submit. And this is actually important. The NIAID has in a number of its different epitope discovery contracts stipulated actually that the recipients of the funding should submit their data to the IDB. And this format is actually conducive to submitting also negative data. We encourage people to submit negative data, and negative data is important because it is important to know whether, if there is no data regarding one particular region, for example, a protein, is that because that the region has never been tested, or has been tested but has been found to be negative? And so that's important information, especially also in the context of training bioinformatic algorithms and so forth. To have negative data is very, very important. And it's about several hundred submissions as of today. And this large-scale epitope submission make up of a significant fraction of the data contained in the IDB. There is a submission process that is well established and validated that involves the investigator contacting the IDB, and then there is automated validation, and a curator is assigned to work with the prospective submitter to make sure that the data is formatted in a way that is consistent with what is in the IDB. And once the submission is approved by both the IDB and the submitter, we don't necessarily release it right away. We understand that in some cases, the submitter might want to hold off making the whole data publicly available until, for example, the publication is accepted and published, so that they don't preempt and steal the thunder of the publication. So this is how the IDB looks like. The page here has a pane where you have the summary of how many epitopes and references and assays have been defined. And then there is a main section that allows you to search for specific the database by specifying discrete parameters, such as what is the structure of the sequence of the epitopes, which organism or antigen it comes from, which host, what type of assays are we looking at, whether T cells, B cells, MHC binding or elution. Do we want only positive assay? Do you also want to look at negative data, MHC restriction, what kind of disease state may be associated with the epitope, and so forth. So, for example, let's pretend here, let's run an example. We want to look at epitopes from hepatitis B. And particularly, we want to see what is known about hepatitis B epitopes in humans. And in this particular example, I'm just going to restrict the search to T cell epitopes, and for which there was positive data. And so if we do that, that's what you get. The banner here reminds you what are you searching for and what are the results related to. And if you do that, you will see that in the scientific literature, there are 471 different hepatitis B described epitopes, T cell epitope in humans derived from 5 different antigens, from 150 different references. And there are 1,500 different experiments linked to that, and over 3,000 different receptors. And I'll get back to that. But in this particular first slide, I'm showing you the what you would see if you are looking at the data displayed under the banner of the epitopes. So here you have all the list of the different epitopes, and you can actually drill down. You can click on the epitope detail, and you will see this epitope summary that tells you what is this epitope, which is a well-known A2 restricted linear epitope, and you see has the data that is available in terms of MHC binding assays, how many times this peptides was tested, and how many times was it found to be positive. Was it tested also for B cell activity? Was it tested for T cell activity, and what assays, and what is the rate of positivities, and so forth. Now, if we go under the antigens banner, we see the data defined and sorted by the different antigens. And in this particular case, for example, we want to look, say, at the envelope data, the S protein data. Now, as I was mentioning earlier on also, different references may have information related to the same epitopes. And so, in fact, this is very good because it's important when a particular structure has been tested by different labs and in different settings, and it's important to understand what is the overall gestalt of data is available relating to one particular epitope or one particular antigen. So how do we aggregate this? We have developed a application, which also it's a subject of a peer-reviewed publications, where we collate all these different data and essentially calculate the epitope density, the density and frequency by which each particular residue was reported to be positive in different assays throughout the scientific literature as present in the curated data. And so you see here this approach defines, either in terms of response frequency or epitope assay counts, some regions that are particularly dominant in terms of what has been described here in terms of, for example, class 2 assays here. I've changed purposely here. Well, the first one was an example of a class 1 restricted epitope. Here is the class 2 reactivity, which is important, obviously, in the context of developing an antibody response. So this data can perhaps be integrated with the data in terms of antibody responses and what's induced by vaccination and so forth, and one can do additional studies. And this is also available as a standalone tool. If you go into the assay tab, then you will have, for example, for this particular epitope, you'll see that this particular epitope has been tested in different assays, for example, chromium or proliferation, chromium release, and you will find for each time this epitope was tested, what was the outcome, and actually what was the reference in which this experiment was described, along with additional information. Finally, very importantly, recent year has been really seeing a blossoming of information related to the sequencing of TCR and BCR data. And so we find this here, also curated in the IDB. And last but not least, you have also the information related to the actual listing of the different references in which one particular associated with one particular query. So quickly, another example of query here, we're looking at influenza. And if you do a very broad search, just any influenza epitope, you will get 10,000 different epitopes. If you include both positive and negative assays, and just anything is known as influenza. But if you narrow it down and say, no, I just want to see only positive assays, now you're down to 5,000 different structures. And you can now say, I want to only look at instances in which there is actual either elution or MHC binding data. And now you see that you're filtering down to approximately 2,000 epitopes. Or alternatively, if you were just looking at T cell data, also it's about 2,000 different epitopes. You can further it down and say, okay, yes, I want to look at influenza data, T cell data, but I want to limit myself to just mouse model and data. And you're now getting about 1,000 different structures that have been tested and for which there is data, and all listed here. You can further drill down and select the specific assays. And so in this particular application, I'm might be, for example, interested in say, well, what data is there for T cell epitope that have been shown in a model system to be protective? And so I can select this particular assays where the epitope was associated with decreased disease or was an adopted transfer, protection from a challenge, and so forth. And so if I hit search, now I zeroed in from thousands and thousands of initial records to about 40 different epitopes that have been defined as T-cell epitopes experimentally in mice associated with an in vivo effect. And so this shows the power of drilling down in the IDP. While all these different search strategies are designed to facilitate for the user to get to what they want, conversely, we are very happy to provide different export functions. And here you see the screenshot of how you can do that. And there's all sort of different exports format that are available in the IDB for people to download the entire database or particular queries or different aspects and be able to analyze them on their own. There are a number of help functions integrated throughout the website. Here, for example, if you look at the epitopes, where you find this little question marks, is usually something that you can, it's a sign that you can access more information regarding that particular pane or tab, and there are specific help resources that are accessible under the help function, and a solution center that you can access throughout the IDP. And with that, I am done. Thank you very much.
Thank you so much, Alex, and perfect timing. Thank you for that great overview of the database.